Now, let me first start with the immune complexes. Immune complexes, or antigen-antibody complexes, I told you, are the lattices of antigen and antibody. Now, these antigens forming immune complexes must be polyvalent. What does this polyvalent means? That each antigen molecule must be able to bind more than one antibody molecule. Now, for immune complexes to develop, antigen must be present long enough to elicit an antibody response. Immune complexes usually form when the antigen is in slight excess of antibody. So, in the last part, while introducing you to the immune complexes, I mentioned the three sources of antigens that were infectious antigens, innocuous environmental antigens, and autoantigens. Now, I will dig into their details one by one. So, follow me. Well, the first one are infectious antigens. Most infections are short-lived and are controlled by the immune response. Even in such rapidly controlled infections, immune complexes may cause hypersensitivity, such as after streptococcal infection. Infections such as hepatitis B are not always controlled and can cause sustained high levels of antigen in blood, called antigenemia, resulting in more chronic immune complex disease. Secondly, we have innocuous environmental antigens. These harmless environmental antigens can elicit an immunoglobulin G, or IgG, response if they are small enough to enter the tissues. A good example is fungal spores, which cause the localized immune complex disease known as farmer's lung. Drugs are also environmental antigens and sometimes cause localized immune complexes, such as the Arthus reaction. Drugs can also cause circulating immune complexes. That leads to a disorder referred to as serum sickness. I will discuss both of them in a while. Now, the third type was autoantigen. Well, autoantigens can only cause immune complex disease in the presence of autoantibodies, an example being DNA as an antigen in systemic lupus erythematosus, or SLE, which is the most prevalent immune complex disease. Normally, innate immune system mechanisms are responsible for clearing DNA. Now, if there's a defect in innate immune system, when cells die, DNA is released into the circulation. So, DNA that is not rapidly cleared can elicit an antibody response. So, these were the antigens of immune complex from three different sources: infections, environment, and self, the autoantigens. Well, I told you in the very start of the video that in immune complex-mediated hypersensitivity reactions, or Type III, you can say three things are stuck together: the antigen, antibody, and the complement. We have discussed the first one, the antigen. Now comes the antibodies. Now, immune complexes will form only when the ratio between antigen and antibody is exactly right. At low levels of antibody, each antigen molecule binds several immunoglobulin molecules, like this, and small complexes are formed. Whereas, when antibody and antigen levels are approximately equal, or antibody levels are slightly in excess, large complexes are formed, like this. On the other hand, when antibody exceeds antigen, again small complexes form. This one. Now, during an infection, at the point where antibody levels are increasing, the immune complexes will be produced very transiently, and these immune complexes rarely become persistent during infections, and do so only when the infection cannot be cleared by antibodies, like hepatitis virus infections. Now, after discussing the antigens and antibodies the third component is the complement. And I mentioned previously that complements help in the clearance of immune complexes. This clearance is necessary, otherwise, immune complexes will cause diseases. Now, the question that pops up in our mind here is that how does the complement clear the immune complex? Now, there are two ways by which complement helps in clearing the immune complexes. Well, it does so, firstly, by the breakdown of large soluble complexes, and secondly, it involves the transfer of the immune complexes to phagocytes by complement receptor 1. Now, let's see how this works. So, the first one, where complement breaks down the large soluble complexes. Now, immune complexes of antigen and immunoglobulin contain high numbers of immunoglobulin Fc in close proximity. Fc, if you remember, is the constant region of antibody. It binds and activates the complement C1 through the classical pathway. We have described the classical and the alternative pathways in detail in our video on the complement system. So, for a better understanding, do watch that video. Well, small molecular components, especially activated C3, are produced through activation of the complement pathway. Now, the C3b molecules insert themselves into various places in the immune complex and break up the lattice of the immune complex. Now, complement receptor 1, or CR1, transfers complexes to phagocytes. The red blood cells transfer circulating immune complexes from tissues and blood to the phagocytes of the liver and spleen, because erythrocytes, or RBC, express complement receptor 1, or CR1, on their surfaces. Do remember that CR1 is the receptor for C3b. Immune complexes bind to the complement receptor CR1 on red cells through C3b. The complex then circulates through the liver and spleen. Here, macrophages also have CR1 along with the Fc receptors, or FCR. FCR is a family of receptors found at the surface of many cell types that bind to the constant region of antibodies, called Fc region. Most Fc receptors bind much more efficiently to antibodies that form part of an antigen-antibody complex, thus ensuring that free antibody in serum does not fill up the receptors and interfere with their function. Well, the macrophages bind to the complexes through both receptors, CR1 binding to the C3b, and FCR binding to the Fc region of antibody, forming immune complex. Now, the macrophages engulf the immune complex, through phagocytosis, removing it from the erythrocyte surface without destroying the RBC, and then degrade the complexes. Even larger aggregates of particulate antigen, such as bacteria, viruses, and cell debris, can be coated with complement, picked up by erythrocytes, and transported to the spleen for destruction. This mechanism is highly efficient and can entirely remove immune complexes from the circulation in a few minutes. Now, what causes the failure of these clearance mechanisms? We know that excess of everything is bad. So, obviously, these mechanisms can be saturated in some situations, where there is excessive, ongoing production of immune complexes, like in antigenemia resulting from chronic infection, and hence, no more clearance occurs. Also, some individuals lack complement. Patients with deficiencies in the early components of complement, C1, C2, and C4, can result in the classical complement pathway not being activated properly and immune complexes not being cleared effectively. Because they do not become tagged with complement, these patients also suffer tissue damage as a result of immune complex deposition, especially in the kidney. Therefore, Lack of clearance of immune complexes will again lead to immune complex diseases. So, here we reach the end of this part, but not the video. Here, we discussed about the different components of immune complexes. I told you about the 3 different sources of antigens, antibodies in immune complexes, and lastly, the complement and its role in the clearance of immune complexes. Now, we are going to discuss what happens if these immune complexes could not be cleared. So, this part is a must-watch.